Shout out to the cytosol.
Glycolysis is a sugar splitting process. It does not need the oxygen to progress, just a hexo sugar to finesse. It all starts off with 2 molecules of ADP. You know me, now let me be. Phosphates change glucose to fructose and create a biphosphate. They call it phosphorylation, the reincarnation of 2 lysis-divided nations.
Cellular respiration is an aerobic process, not meant to impress, simply set for success.
The production of NAD is oxidized, the creation of NADH is compromised, the energy that is drained transcends, and the phosphates are added for friends. Enzymes remove the phosphate to conglomerate with adenine dye. The end result is 40 adenine tri, 2-pyruvate, and 2 NADP for supply.
Cellular respiration is an aerobic process, not meant to impress, simply set for success.
With O2 present, the pyruvate is actively passed to the matrix, followed by hat tricks, CO2 with the latest greatest waste. In result, the acetyl group is oxidized with NADH, so acetyl-CoA is created and cultivated. When acetyl-CoA is present, the Krebs cycle's intent. The summation of 2 divided nations creates citrate, a 6-carbon compound meant to oxidate. Meanwhile, NAD plus is reduced and then the NAD produced. Makes sense?
Cellular respiration is an aerobic process, not meant to impress, simply set for success.
Now we're left with the 5-carbon compound, and the process comes around. The 5-carbon compound oxidates to create 4 carbons in a reduced NADH. The next step is complicated, but let me elaborate. NADH is produced, and yes, we are back to that. The coenzyme FAD is reduced to FAH2, and ADP is reduced to ATP2. Then we repeat, featuring oxaloacetate. The Krebs cycle is an aerobic process that creates 4 ATP for you and me.
Oxidative phosphorylation is a metabolic pathway. Nutrient release is forming ATP. About to go the metabolic way. Highly efficient. Phosphorylation don't need a permit to do its work. Other processes only about as useful as a hotel desk clerk. Glycolysis just can't compare, and phosphorylation just don't care. Electron transport chain. It's only step 1 of this oxidative process. ETC ain't acquainted with ATP, but knows that electrons love the progress. NADH and FADH2 start the movement. Electrons go down the lane. This is all taking place in the mitochondrial inner membrane. Protein after protein receive electrons, lead straight to loose protons. They flow through the membrane like cold weather through your long johns. Chemiosmosis is step 2 of oxidative phosphorylation, a proton gradient ATP synthase. It's an all-important affiliation. The protons produced in ETC move back across the inner membrane. That energy powers ATP synthase. We all know this enzyme is just insane. ATP synthase binds ADP with an inorganic phosphate molecule. The end result is 34 molecules of ATP. It's our metabolic fuel.